In the natural world, we think we know the basic rules. A cat has kittens, an oak tree makes acorns, and a queen ant produces ants of her own species. But biology is full of rule breakers, and today we're talking about one that has torn up the very textbook of reproduction. This is the story of an ant that reproduces by quite literally copying another species. It's a tale of genetic theft, cross-species cloning, and a biological entity that forces us to question what a species even is. This is the astonishing Iberian harvester ant, Messor ibericus. For over a century, a core principle of biology has been that you give birth to your own kind. Hybridization, the mating of two different species, happens, but it's usually a dead end, creating sterile offspring like mules. It's an exception. But in the sun-drenched landscapes of the Iberian Peninsula, scientists have found something so unprecedented it needed a new name, xenoparity, which means to give birth to the foreign. Queens of the ant Messor ibericus have evolved a way to create and sustain members of an entirely different species, the builder-harvester ant Messor structor, inside their own nests. They do this to ensure their own lineage survives. It's a form of what scientists call sexual domestication, where one species has hijacked the reproductive ability of another. The result is a colony that's a mosaic of life, a purebred queen of one species, cloned males of another species, purebred males of her own species, and a sterile workforce of hybrids. The journey to this discovery didn't begin with a flash of insight, but with a nagging puzzle. Genetic surveys of harvester ants across southern Europe kept showing something strange. In the colonies of the Iberian harvester ant, Messor ibericus, all the worker ants were hybrids. Their genes were a 50-50 split between Messor ibericus and another distinct species, Messor structor. These two species are not close cousins. They split from a common ancestor over 5 million years ago, making them as genetically distant as humans and chimpanzees. Interspecies mating isn't unheard of in ants, but it usually happens where the territories of two species overlap. And indeed, in some parts of the Iberian Peninsula, both these species coexist, so it seemed plausible that Messor ibericus queens were simply mating with Messor structor males. But the mystery deepened when these hybrid-filled colonies were found in regions where Messor structor was completely absent. On the Italian island of Sicily, for example, biologists confirmed there were no native colonies of Messor structor. Yet, they found thriving Messor ibericus colonies whose workers were the same telltale hybrids, carrying the genes of a species that lived over 1,000 kilometers away. How could these queens produce hybrid offspring if there were no local males of the other species to mate with? It was a logistical impossibility. The queens weren't finding foreign males. They were somehow bringing them along for the ride. To solve this enigma, scientists brought the colonies into the lab. And here, under controlled observation, the truth revealed itself. From a single Messor ibericus queen, in a single nest, emerged two completely different types of male offspring. One group of males was exactly as expected, hairy, genetically pure Messor ibericus. The other group, however, was genetically and physically identifiable as Messor structor, nearly hairless, and belonging to an entirely different species. A single mother was giving birth to sons of two different species. This was the smoking gun. The proof that the queen wasn't just mating with foreign males, she was producing them herself. She was, in effect, cloning males of another species. This single observation shattered conventional biology. The queen carries the genetic blueprint of the Messor structor male with her, not as part of her own DNA, but as a tool she can deploy. She doesn't need a wild population of Messor structor, because she can generate her own supply of males from that species whenever she needs them. This is xenoparity, the first time a female of one species has been seen to clone members of another. So how does this factory of genetic theft actually work? The reproductive life of a Messor ibericus queen is a masterclass in genetic manipulation using 3 distinct pathways. First, creating the hybrid workforce. This is the whole point of the system. For reasons we're still exploring, it seems that if a Messor ibericus queen mates with a male of her own species, her female offspring can only develop into new queens. A society of only queens is a non-starter. You need workers to forage, tend to the young, and defend the nest. To solve this, the queen must mate with a Messor structor male. She uses sperm from this mating to fertilize her eggs, and these eggs develop into the hybrid, sterile female workers that form the backbone of the colony. Second, producing the cloned foreign males. This is the revolutionary part. To ensure a continuous supply of Messor structor males, the queen performs a remarkable genetic trick. She takes one of her own unfertilized eggs, and in a process not yet fully understood, she essentially erases or ejects her own nuclear DNA. This empty egg becomes a vessel. It is then fertilized by sperm she has stored from a previous mating with a Messor structor male. That sperm's DNA then becomes the sole nuclear genome for the developing embryo. This process is known as androgenesis, meaning male origin. The result is an offspring that is a clone of its father, a genetically pure Messor structor male. The only trace of its mother is the mitochondrial DNA, which resides in the cytoplasm of the egg. These males are, for all intents and purposes, members of another species, produced by a foreign mother. Third, reproducing her own lineage. To continue her own species, the queen also produces males of her own kind, the purebred Messor ibericus. She mates with one of these sons and uses his sperm to fertilize her eggs. This specific genetic combination results in the development of new, purebred Messor ibericus queens. These virgin queens eventually fly off to start their own colonies, carrying this entire astonishing reproductive toolkit with them. The queen acts as a central processing unit for a complex reproductive factory. She produces purebred sons of her own species and cloned sons of another. She mates with both, using the sperm to create either new queens for her lineage or hybrid workers for her colony. How could such a convoluted system even evolve? The leading theory points to obligate sperm parasitism. In many harvester ant species, queens are known to mate with males from other lineages to produce their workers. It's thought to be a way to avoid genetic conflicts that can arise from inbreeding. It's likely Messor ibericus started down this path, becoming dependent on Messor structor sperm to create its workforce. At first, this was a major limitation, tying them to areas where Messor structor also lived. The evolutionary game changer was the development of androgenesis. The moment the first Messor ibericus queen figured out how to not just use foreign sperm, but to clone the male that produced it, she effectively captured his entire genetic line. This transformed the relationship from dependence to sexual domestication. Just as humans domesticated wolves into dogs, the Messor ibericus queen has domesticated the Messor structure genome. She brought the source of the foreign sperm in-house, creating her own private, self-renewing supply of mates. This freed the species from its geographical chains, allowing it to expand into new territories, like Sicily, where its mate species doesn't even exist. The result of xenoparity is a colony that defies our categories. It is a two-species superorganism. The colony functions as one, yet it's built from the parts of two species that diverged millions of years ago. This challenges our very concept of biological individuality. Is the Messor structor male, born from a Messor ibericus mother, truly a Messor structor? Morphologically, he's a little different from his wild counterparts. Genetically, his nuclear DNA is pure Messor structure, but his mitochondrial DNA comes from a Messor ibericus mother. Functionally, he is completely dependent on the Messor ibericus colony. He can't reproduce with his own kind because there are no Messor structure queens around. He is an integral but alien component of the Messor ibericus reproductive cycle. One entomologist suggested these males shouldn't be classified as hybrids or even as Messor structor, but as an essential element of the Messor ibericus population itself. They are living, breathing reproductive tools, owned and propagated by another species. While the case of Messor ibericus is spectacular, it's part of a broader world of bizarre ant reproduction. Many ants have evolved forms of asexual reproduction, or parthenogenesis, to adapt to their environments. Parthenogenesis, or virgin birth, is fundamental to ants, bees, and wasps. Their sex determination system, haplodiploidy, means males typically come from unfertilized eggs. But some ants take it further. The clonal raider ant, Ushirea biroi, has done away with queens entirely. Every individual is a female that reproduces by cloning herself, creating an endless stream of identical daughters. They function in synchronized cycles of foraging and egg-laying, expanding through collective cloning. Then there's the European desert ant, Cataglyphus cursor. Here, the queen produces new queens asexually, cloning her royal lineage. But the sterile workers, who need to be strong and disease-resistant, are produced sexually, giving the colony genetic diversity where it counts. It's the best of both worlds, genetic continuity for the queen line and genetic vigor for the worker force. But perhaps the only case that rivals Messor ibericus in complexity is the Japanese ant, Volenhovia emerii. This species uses a dual cloning system. New queens are clones of their mother, produced asexually. Workers are produced sexually. But the males are clones of their fathers. When a queen uses sperm to create a male, her own genetic contribution is ejected, and the resulting male is a clone of the male she mated with. This creates a complete separation of the male and female gene pools. The female lineage passes clonally from mother to daughter. The male lineage passes clonally from father to son, using the female as an intermediary. The two gene pools only mix to create the sterile workers. The discovery of xenoparity in Messor ibericus is a profound reminder that the natural world is always more inventive than we can imagine. These ants have broken a fundamental rule of biology, evolving a system of cross-species cloning and sexual domestication that sounds like science fiction. They turned another species into a living, renewable resource. This strategy, alongside other clonal ants, paints a picture of a group of organisms that are masters of evolutionary innovation. Ants have repeatedly bent and broken the rules of reproduction, developing systems that are elegant, bizarre, and perfectly suited to the complex challenges of social living. The story of the Iberian harvester ant is not just a curiosity, it's a fundamental lesson in the power of evolution. It shows that life is not bound by rigid laws, but is a fluid, dynamic process, capable of producing solutions that seem almost unimaginable. The queen that gives birth to two species has given us a stunning new example of nature's ingenuity, and ensures that the humble ant will continue to be a source of wonder and scientific revelation for years to come.